All right, so hi, I'm Tom. I'm from scalableminds, and today I'd like to talk a little bit more about webKnossos and our software tools and solutions for working with volumetric datasets, in particular large-scale microscopy datasets, EM and the like. And as part of that, I would like to share a little bit of a case study later on, just to see how all of the offerings that we have fit together. To quickly motivate all of that a little bit, and I'm sure some of you are already facing some of these issues that we and our partners are already dealing with. So data is getting bigger and bigger. The kinds of image data sets that we can acquire these days with modern microscopy, especially for 3D data sets, volumetric data sets in the EM domain. They're getting bigger and bigger. Whereas a few years ago, we thought that handling stuff like a full scan of the Drosophila brain is already pushing the boundaries. We now have some of the researchers out there, especially in the field of neuroscience, that are looking even beyond that and are working towards imaging a full human brain in the future. And the question that comes with it, that is, how do you deal with all of that data? How do you analyze it? How do you visualize it? This is where we specialize in providing software solutions for these kinds of problems. Perhaps from a little bit more of a different angle is then the question, what do I mean when I talk about large image data sets? And I think this is very different for each one of us. Sometimes it's already big if I can't email it to my colleague when I want to share it. Sometimes it's big or too big if I cannot open it on my laptop any longer. So here we are perhaps in the gigabyte scale already. And when I want to start doing any sort of analysis, my data sets might already be too large to fit on a single workstation computer. So really what we're looking or what we're seeing is that more and more life scientists are hitting the limits of what fits on a single drive, what fits on a single computer. And we need to look beyond that for solutions. So this is a little bit where webKnossos fits into the picture. So webKnossos, as Claudia already introduced a little bit, is our web platform for visualizing, sharing, and annotating large image data sets. It is designed in particular for volumetric data sets. Our background is in computer science, but most of the scientific partners that we're working with typically come from the field of neuroscience, so has a little bit of bias towards that, but generally speaking can be used for micro CT, MRI, CT scans, and other modalities just as well. In addition to webKnossos, our company has another tool called Voxelytics, which is more our AI software tools for the analysis, and I will talk a little bit more about that later on. So what can I use webKnossos for? As I mentioned, it's a web platform for visualization, annotation, and sharing. So one of the earliest features and the most helpful things is that it was built in order to view these large-scale data sets and stream data dynamically, directly from your storage servers. So there is no need to copy any data around any longer. There's no need to send around hard disks. We can directly stream any of the large-scale data sets from an scientific storage server on your institute, on your campus, at your company. People don't need to install anything. It's all web-based. So you basically can share a web link, and people can very much interactively explore data sets without much complications there. So in this particular scene that we're seeing here, we are going through several terabytes of data without much hiccup. On top of that, I mentioned that you can do annotations. So webKnossos can do several sorts of annotations. We have volume annotations, so you can just brush in individual objects, individual cells. We see that being used in some cases as the analysis itself. So if you only want to mark a few cells manually, that's fine. But in other scenarios, we use this for gathering training data to then train AI models, machine learning models for doing automated analyses. Additionally, we support these kinds of skeleton annotations that we see in the lower right. So basically placing nodes in the data set and then connecting them. This is mostly for, or that fits very well for reconstructing large spanning structures such as neurons and the like. And for this particular purpose, a few years ago, we published this as well in Science. We developed what we call the flight mode. So the idea behind that is, imagine that you're sitting in the cockpit and you're sort of flying through the data set here, and it takes a little while to get used to this kind of annotation, but then you're really quick if you want to make a lot of these kinds of large-scale skeleton annotations. So at this point, I sort of want to jump in and do this a little bit more interactively and show you around. If you want to follow along, webKnossos is free and open source. So you can just go to webknossos.org or tinyurl.com/webknossos, and this will also bring you to the same data set for you to explore. So here we see the basic interface of webKnossos. I've loaded in an EM data set already. We have the different viewports along all of the major axes. And in this particular case, it's from mouse cortex. So EM data, serial block phase data. And as I zoom in and out, webKnossos dynamically loads the data from the data center in the cloud, or in this particular case, actually from a server of the Max Planck Institute. And on top of the raw EM data, we can put additional information. So we have all sorts of layers that we can add. So here we did a full segmentation of this particular piece of a data set to really reconstruct all of the neuron pathways and connections between them. And as we see down here in the bottom right, I already preloaded this. We support creating 3D meshes and visualizing these segmentations as 3D meshes. So you can really get a good view of your data and your reconstruction and make sense of it. Not logged in. Well, you can sign up for a free account on webknossos.org if you want to get started for free. There's a little bit of several gigabytes of storage available for everyone. And or alternatively, you can do a custom installation at your institute. So on top of some of the annotation features that I mentioned earlier, especially if you want to do large scale research, or if you are in a group of, in a research group, you want to collaborate on these things, and webKnossos supports all of this. So we have project management features, we have group annotations features, we have permissions and a rights management system, we have a task system that allows you to organize your data annotation as projects, which then can be distributed to either a number of students that you may have, or your colleagues and so on, and you can monitor the progress of all of these projects, and this enables you to really work on some of these larger scale reconstruction projects. In cases we're not interested in doing all of this manually yourself, we also do offer that as a service where we will annotate data for you if that makes it easier. So at the end of all of this hard work that you put in there as a researcher, the best part of it is typically when you're at the point where you can actually publish your work. And again, here webKnossos helps. We found that a lot of our users like to just include the links directly to the data within their papers. So here we have another paper from Science published last year, where both the annotations and the raw data are directly in the paper. You can jump right where the author wanted to highlight features, and it's very seamless for anyone who's going through the paper on how to access the data and how to explore it and analyze it for themselves if they're so inclined. And if you want to, or on top of just the web interface, we provide Python libraries for you to integrate all of that into your own workflows. So that enables you to either download the data, to upload new data sets to webKnossos, to combine it with existing workflows that you might already have, especially if you're doing some sort of automated reconstruction. So a lot of flexibility there, and you're not stuck with any of your annotations in webKnossos. You can download it, bring it into your own programs, and really mix and match so that it makes sense for you. And for some of the community data sets that we have, we also feature them on webknossos.org. We have a little bit of a gallery there. So these data sets are available there, very similar to what we heard earlier on the other repositories sponsored by EMBL. This is just a small selection of typically large-scale EM data sets that either you can use for your own research or that we feature for the community as part of publications. So with that, with this little overview of webKnossos and some of the features there, I want to turn my attention to a case study. So we have very good timing on that. Just this week, together with the Max Planck Institute for Brain Research in Frankfurt, we published a paper on connectomic comparison of mouse and human cortex. And their particular field of research is in connectomics, so the reconstruction of neurons and their interconnection and so on. At the end of the day, it brings a lot of interesting challenges on data handling and reconstruction, and I want to walk you a little bit through how our software tools here help with that. Interestingly enough, this morning I realized that even Spiegel, one of the larger German news journals, picked up on this, so good timing. So in this particular study, the researchers did a screening, a comparison of 8 different data sets, and the samples were taken from mouse, macaque apes, and humans, and all processed and imaged with serial block phase imaging. And then they applied the voxelytics procedures for automated segmentations and reconstructions to really get a full reconstructions of all the cells and of all the neurons in each of the data sets to then do comparative analysis. And while going through this step by step, the researchers were facing various problems that I'm sure some of you are familiar with. So especially when working with volume EM data, the first thing that you have to do is align the data, make sure that you stitch together the individual slices that you imaged along the Z-axis. And here we offer the routines for robust alignment as part of our voxelytics offering. The next challenge that we had to face is how do we do the automated neuron reconstruction? So we used machine learning and automated segmentations with the voxelytics framework. We collected all of the training data in with webKnossos, trained it, and we're able to, well, we're able to reconstruct neurons on the detail level that you see here on the right-hand side. This worked great for most of the mouse data sets that we already had some of the pre-trained models available, or that parts of our work were focused on initially anyway. But then when we turned our attention to macaque and human, we were running into some issues where the morphological details and some of the staining within the data sets looked different from what we were observing in mouse. So we had to retrain data. And again, here we use the annotation features within webKnossos to create new training data sets. So we went back to the drawing board, labeled more data, distributed this out as tasks to several people, and we're able to very quickly annotate several large bounding boxes with dense volume annotations and use that for retraining. On top of that, to make the neuron reconstructions as error-free and as correct as possible, we include additional biological constraints in our agglomeration strategy. So some of the features that we take into consideration are nuclei. So we have dedicated classifiers for identifying nuclei, just to prevent two processes from being merged together that that already are connected to a nucleus. There can only be one. And we also do type annotations, or we infer types of each cell. So typically exons, dendrites, astrocytes, and so on. On top of that, to also prevent any unnecessary mergers. Both of these systems are based on separate classifiers, once again. An interesting feature that we have within webKnossos is what we call ID mapping, or working with agglomerates. So typically with either or with these kinds of segmentation projects, the initial outcome from a classifier is in many cases an over-segmentation. So it's broken down in smaller pieces, and then through a series of manual or automated steps, you pick up the pieces once again. And within webKnossos, we have a feature to show different states of that, so you can more or less interactively load in the agglomeration results of these in more individual pixels. Sometimes this is also called like working with a super voxel graph and so on. And recently we started offering proofreading tools for all of that. So obviously whenever, or in cases where you do work with automated reconstructions, it can happen that there are errors in there. So we observe merge and split errors in our segmentations, and we now have tools to fix this on the fly. So here we see me splitting two neighboring cells that used to be fused together, and by deleting or the connection in between them that the program estimated, we can split them into two separate parts as seen on the bottom right-hand side. And once we're happy with most of our reconstruction work, we then have the capabilities to visualize all of that as 3D meshes in webKnossos itself, and the capabilities to download that and either use that for interesting figures, use it for further proofreading. Sometimes it's easier to spot mistakes in 3D, or use it for public relations work and so on. And this brings me more or less towards the end of this pipeline. Once we've reconstructed all of the individual neurons, we then can move on to what we call the connectome reconstruction, where we sort of figure out who's connected to who, which neurons are interacting with one another. And the last missing pieces for that is synapse detection. So we have yet another classifier that is taking care of that to both identify synapses within the data sets and label post and presynaptic partners. And with that information, we have all the pieces of the puzzle together to then reconstruct the full connectome. We have measures in there for evaluating the results of a classification within webKnossos. So a very versatile suite of software tools. So one of the results may look like this, a big matrix of the interconnections, but it's more fun if you want to interactively look through that. So we have what we call, or we can bring in all the synapses for analysis into webKnossos, where you can interactively load them for each of the neurons, and then find neighboring connections, filter, and so on. So that brings me to the last part of my presentation. What's next on our agenda? So as mentioned earlier by others, we're also working towards supporting the SAR standard. Most of the data sets within webKnossos can be exported through a SAR interface, or the other way around. We can load SAR data from cloud storage and local storage as well. So here you see me opening a data set that is located in a webKnossos instance, but it's loaded in a third-party tool that is also compatible with SAR. Also coming up is the tighter integration of webKnossos and our AI toolkit, Boxalytics. So right now, the two of them are thought together and work very well together, but they are executed separately from one another, and we're working actively into incorporating the two more and more, so that you can launch some of the AI segmentations that I showcased here directly from within webKnossos. As mentioned earlier, you can get started with webKnossos if you want to try it out on webknossos.org. That gives you an opportunity to upload your own data sets or bring it in from a neuroclancer link and so on, and in the future also from SAR. And there's also the opportunity to host it yourself if you already have a large repository of data sets, and it makes more sense to have it on premise. So that's all from me today. Thank you for listening, and I'm open to any questions you might have.